Hello everyone, my name is Jasmine Kaur. I'm from the University of Indianapolis, and as a Hunter College SPUR student, I was able to conduct research this summer on how prostaglandin J2 alters the amyloid precursor proteins glycosylation and trafficking and its relevance to Alzheimer's disease. Alzheimer's disease is a global problem. 35 million people worldwide are affected with this neurodegenerative disorder, and that is initiated by neuroinflammation, and which eventually leads to various cognitive and behavioral deficits. There are various pathological hallmarks of Alzheimer's disease. One of them is the key formation of beta amyloid plaques, and these plaques are the result of incorrect APP proteolytic processing. And these plaques are very neurotoxic, and which eventually cause cognitive dysfunction within the brain. Another hallmark is the presence of neurofibrillary tangles, which are aggregates of the tau protein. And both of these combined lead to the loss of neurons and synaptic connections. Epidemiological research on NSAIDs use say that cyclooxygenases and their prostaglandin products may play a critical role in Alzheimer's disease. Because when the beta amyloid plaques start forming, the use of NSAIDs is no longer effective because they actually have an inhibitory effect on the chronically activated microglial cells. As you can see on the left, this is a normal neighborhood of a neuron with its supporting neurons as well, which are called the microglial cells. However, in the neighborhood in Alzheimer's disease, the microglial cells are highly activated, which release pro-inflammatory signals, and one of those signals is prostaglandin J2. And as you can see, compared to the processes on the left side, the processes on the right are actually deteriorating, which cause the loss of synaptic connections as well. Prostaglandin J2 is an endogenous neurotoxic product of cyclooxygenases, and cyclooxygenases are key enzymes in the inflammation cascade. And as you can see on the scheme on the right, neuroinflammation causes glial cells, microglial and astrocytes in particular, to release prostaglandin J2. And this causes an intracellular increase in prostaglandin J2, and this has a positive feedback loop on the enzymatic activity of the COX-2 enzyme. And this positive feedback loop causes a transition from acute to chronic neuroinflammation. So the question that is proposed is that what is the effect of prostaglandin J2 on the APP glycosylation and trafficking? Some background about the amyloid precursor protein is that it's a ubiquitous receptor-like transmembrane protein that undergoes glycosylation as one of its post-translational modifications. The APP's physiological functions include synaptic transmission, synaptic plasticity, and neurite growth, and overall it's highly neuroprotective. And it plays a key role in Alzheimer's disease pathology because of its incorrect cleavage causes the formation of the plaques. The image on the left shows APP glycosylation. Once APP is translated, it gets N-glycosylated within the endoplasmic reticulum. And N-glycosylation is specifically is when a sugar complex is attached to an asparagine amino acid residue. And this N-glycosylated APP is immature still. It has to be translocated to the Golgi to undergo O-glycosylation, which is a sugar complex that's attached to a serine or a threonine amino acid residue, and eventually then it gets matured. And as you can see, APP trafficking, the image on the right, it's packaged within vesicles. After it gets O-glycosylated, it goes through axonal transport, and eventually it gets transported to the presynaptic terminal. So our experimental model in our experiments was rat cerebrocortical neuronal cultures. We were able to obtain day 18 embryos, and we were able to isolate the cortices specifically because they are involved in higher functions of brain activity. We used an enzyme called papain in order to disperse the cells in culture. The cells were in culture for 1 week exactly, and we were able to then treat the cells with 10 micromolar PGJ2 for a duration of 16 hours. Pre-existing data from our lab shows that a 16-hour PDJ2 treatment alters APP levels in the cortical neurons. This is a concentration curve representing various levels of PDJ2 compared to DMSO. And DMSO is our control group throughout these experiments because DMSO is an organic solvent and PGJ2 is dissolved within DMSO specifically. And as you can see in the Western blot, at 10 micromolar PGJ2, the full-length APP concentration, it decreases compared to our DMSO control lane. And we use 10 micromolar PGJ2 as our optimal concentration because, as you can see from the cell viability graph, at 15 and 20 micromolar concentrations, there's a decrease in cell viability, so we did not want to kill our cells by using a higher concentration. So we use 10 micromolar throughout. Data also has shown that PGJ2 alters APP glycosylation. The two graphs on the left side show mature APP bands that are shown within the Western blot as well. In the DMSO control lane, you can see that there's 3 specific bands. The top 2 in all these these lanes show N and O glycosylated APP. However, and the last band you can see is the immature APP band, that is N-glycosylated. So you can see that there's a decrease in APP that is O-glycosylated in a concentration-dependent manner, while APP that is N-glycosylated stays consistent, and there's no statistical significance in that graph specifically. So we were able to conclude that PGJ2 disturbs O-glycosylation, which occurs in the Golgi, as I showed you earlier in their image, but not N-glycosylation, which occurs specifically in the ER for APP. So this leads to our hypothesis that PGJ2 disturbs APP glycosylation, and this abnormally glycosylated APP may impair APP trafficking. And if there's a decrease in the normal levels of APP, this can contribute to neurodegeneration because overall APP is neuroprotective. And in order to cause neuroinflammation in our experimental model, we used PGJ2, and we saw APP glycosylation was disturbed, and eventually neurodegeneration occurred. So in order to see the effect of PGJ2 on APP trafficking, we did a immunocytochemical analysis. We used our rat cortical neurons. They were 7 days in vitro. Our control group was DMSO. Treatment was 10 micromolar PGJ2. We were able to fix the cells with paraformaldehyde. We blocked them with BSA and NGS. Our primary antibody for APP was 22C11. And later we incubated them with a secondary antibody. The image on the right, as you can see, is the somas are supersaturated because we wanted to visibly see the processes. And this image comes from our DMSO control group. And as you can see, there's a continuous, clear presence of APP that is shown in red. So we were able to obtain 3 representative images from each of our conditions, DMSO and PGJ2. There's a clear indicative presence of APP in the DMSO that is shown in a continuous manner. However, in PGJ2, there's clusters of APP, and this presence of the clustering of APP suggests that there's an impediment of trafficking that may be occurring within the processes. We were able to do immunofluorescence quantification using an ImageJ software. The location that we used was the neuronal processes, and this is an example of a scheme of how we actually quantified them. We cropped out the processes, and after using our software, we found statistical significance within the number of small particles compared between our 2 groups, DMSO and PGJ2. There was a smaller amount of small particles, lower amount of small particles in PGJ2, indicating that because of those clusters, there wasn't as much small particles as was seen in our DMSO controlling, which is also representative in that scheme. So our conclusions in summary, our ICC immunocytochemical analysis suggests that PGJ2 disrupts axonal trafficking of full-length APP, and future studies include that we want to investigate PGJ2, whether PGJ2 causes accumulation of APP at the ER, which eventually would prevent its translocation to the Golgi, and we would able to conduct this experiment through subcellular fractionation in order to separate the organelles. Alterations in APP glycosylation induced by PGJ2 will have an impact on intracellular sorting and processing and export APP. We want to look into that even further. And overall, the study was very important, and it's highly relevant to Alzheimer's disease because we saw that in the processes there is a disruption that's occurring, and how neuroinflammation causes a deterioration of the processes that leads to cognitive and behavioral deficits in AD patients. So, and I would like to acknowledge my principal investigator, Dr. Pereira, my lab mates Tanika and Jorge for all their help this summer, and which I really enjoyed, and the Hunter College SPUR program and the 2 grants that were used to fund this program. Thank you very much.